So let me just start with one question for all of you, and then we'll open up to the room. I see quite a few questions here. What I found interesting about your careers is that you really, all three of you, did not get to where you are in your research in a kind of straight, direct route, right? I think, we have a case of a surgeon, right, who became a luminary in the field of senescence and genome structure. So, Melike, if I remember correctly, you started as a physicist, right? That's how you get your degree, but your early research was on viruses. And now what we witnessed, some fundamental discoveries in genome structure. And in fact, in your case, you actually started with with microscopy, but you have also made some seminal discoveries related to neuroscience and brain function. So what I'm curious about is, were there any critical events in your life that made you change what you do and succeed in where you are now? And what are they? Let me just give you a microphone.
Why I changed the path? Is a formal answer or convenient one? So, it's so long ago. I don't remember. Maybe I kind of idealize, no? It's just surgery or medics, one by one. Fundamental biology, of course, zero or a little bit bigger. So that was sort of my formal answer, but probably not true, entirely true. And it's partially true. So in Japan, residents were crazy. I don't know now, 20 years ago when I was there, literally crazy, right? It's just over the limit. And then I did the 5 years resident plus some staff. Probably I worked enough amount of one person could work for their life, normal life. So that's my feeling. I still, as a surgeon, still junior, but as a medics, I felt I did enough for probably for life. So then I just start, for some reason, I really wanted to do some research. So I just grabbed the opportunity for the PhD position. There was one PhD position there, happened to be there when I was sort of the timing where I need to move the universities, hospitals, and then I went back to the university and then I take the opportunity. And I just never went back to clinic. So it's hard to make sense retrospectively. Sorry.
Any other life stories?
I mean, I wouldn't say there was one critical moment that made me, I think it was a lot of random chance, a lot of being at the right place at the right time in the right environment. I think there's a lot of that in science. And, I've been asked in the past to give career talks, and I always name it as my windy career through science because it's not a straight path. It's always very windy, and we often don't talk about the reality of that. So I started as a physicist, but I never had any formal training in biology, but I was always excited about and interested in biology. Already as an undergrad, I did research in biophysics, soft condensed matter physics, and slowly over time got interested in applying the physics background, the physics training, and the quantitative thinking to biology. And so when I started my PhD, I was very fortunate to meet my mentor, Huang, and was interested in, at the time, looking at influenza virus using microscopy, single particle tracking. It was really like early days of live cell imaging, and that sounded really exciting. And so I jumped on the opportunity. And yeah, through my own career, it's always been, where the science takes you. And, again, with, for example, chromatin, started as a collaborative work, and I've been very fortunate to meet amazing collaborators from different backgrounds: stem cell biology, mechanobiology, etc., and just collaborating. And those things take you in different directions that can be a lot of fun and exciting. So, I think it's again a lot of chance encounters and meeting the right people and taking the opportunities and following them up.
I actually started with theoretical physics in my undergrad. I was a theoretical physicist working on a quantum theory. And I went to the University of New Mexico because they have a really good quantum information group. I sit in some of these seminars, and I just figured it has no relevance, at least at that time. It was too early. And then I think, okay, something that is a little bit more relevant is biophysics. So I went into a biophysics lab. I don't think during the time I back reflect, I'm much, in a junior stage compared to the other two investigators. But I never thought of what I could do to develop my career. All I think is these are interesting things I want to do. When I was graduating from UNM, I joined Jörg Beversdorf lab as a postdoc. At that time, there's not like, I think this thing will land me a faculty position, but rather I just think for pi, single molecule imaging is very unique. I just want to have my hands on it. And at that time when I joined, there's no plan of publication. It just said, let's try it. If we can make this at least is something difficult. But I think when I work on it for a fairly long time, we find a lot of things we can do to make it whole cells into tissues and all these stuff. And some of our earlier career publications actually come from just testing a scientific camera from a company. Right? The company said, we have a camera, you want to test it? I said, sure, let's try it. And then that becomes one of the first papers. So that's my perspective.
Hey, Huang, great work. It's very interesting that you introduced noise term as part of the information theory to modify or enhance the diffraction limit equation. Can I challenge you for making the noise term more complicated in a way that photon count can be improved by integration? So if you introduce a time term, for example, right, single molecule localization are based solely on, when you say 500 to 1,000 photons, those are single frames. You can combine 100 frames potentially, if it's possible to combine. Will that further modify your equation?
I think that would be a wonderful idea. And one thing I think we found it very difficult to do is to incorporate single molecule blinking into this equation, because they are stochastic and they can overlap and they depend on the performance of algorithms. Right? So something that, if we want to incorporate time, we have to think about all the statistics. That add in another layer of randomness. And currently, we only have very simple randomness with Poisson noise. Something that along the line, what you suggested, might be camera noise, right? That's something we didn't think about. The camera noise is also inversely depend on the time of duration. If you increase exposure time, but you don't increase the camera noise. So something, I think there is a lot of things. This is like, I would say, It's just the first step. And in future, what we really hope is that, if you have a structure you want to resolve, like Maliki, you want to resolve, let's say 20 nanometer features, and you put there maybe a website that you can go in and click, I want to resolve 20 nanometer features, and what type of modality I should use, and what's my photon budget. You click a button, it gives you some suggestions.
That would be wonderful.
I think that would be cool.
Yeah, absolutely.
See, I think there is a question here too.
Thank you, Vadim. So thank you so much, the three of you, for this banquet of good science that we had in the morning. Thank you. I think that my question as an apprentice to the field of chromatin organization, histones, and how the domains form, relates to, it seems that there's a lot of emphasis in the field in terms of histone modifications, more than variants. Is that conceptual because there's a belief in the field that modifications are biologically dominant over the variants, like the variants don't matter as much? Or it's technical because the antibodies suck or you don't have good tools for variants? So what's your perception about this modification versus variant?
You want to take this? I take it. I mean, I don't think one is more important than the other. I think they're equally important. Modifications, it's a combinatorial code in my mind, right? It's not just one modification, it's multiple modifications, and then there's the histone variants. And then, there's the linker histone, the sort of forgotten histone that also has variants and, isoforms and plays each important roles and potentially differing roles in different cell types. So it's a very complex, I think, combinatorial sort of signature that defines underlying function. Why the variants are not studied, that I don't know. I'm not really like a cart-carrying chromatin biologist. Perhaps the lack of resources, reagents is an important one. In the imaging that we do, we rely on existence of good antibodies, for instance, that can differentiate between different modifications or different variants. And when you go to the variants, it's harder to do that because they sequence conservation is high, and it's hard to really detect one over the other one. So that could be potentially a reason why it's much harder to study the variants. But I think they're equally important. I don't know if you have something to add to that.
I think I agree. It's just not investigated enough yet. And the variants could be just the same layer within the same layer, another epigenetic player. Some variants very similar. Lots of them are actually, histones. So many identical variants. Why? It's more to do with the quantitative aspect of that. I found this is extremely interesting as well. Just a little bit beyond your question. But yes, so it's just another complexity which just don't know much about it yet.
So if you have a question, please raise your hand, and then I will give you the mic. There's one thing that I believe is on the minds of quite a few people in this room: AI. Right? We hear about this all the time. What do you think the role of AI is going to be in your own research, your field, or in science in general?
I'll let Huang answer first because he's already using a lot of this.
Yeah, so I would say our investigation or using AI is really like baby step, right? Compared to current large language models and all these different uses. But I think what we're trying to do is trying to do something that human cannot do. It's not about developing like self-driving cars where you can already drive a car. We want to do something that, combining mathematical thinking and physics, because a lot of us are engineers and physicists, we can put those in. And before, it's really complicated for any algorithm to handle. Now we can handle. And to control very complex instrument that, just beyond the human understanding. And this is something that we really intrigue us, and trying to go beyond the human limitation. Because in some of these applications we have, if we apply it to a method that can be also done in classical manner, and those classical computation methods in some time can be speed up by parallel computation as well. So there's a question whether do we want to go this route or that route. So that's usually our initial thoughts.
Yeah, I mean, I think that for our work, where we do a lot of microscopy and imaging and image analysis, AI is going to play probably a very important role, and I think it's going to be the future of image analysis. Again, as Huang mentioned, what we do right now with machine learning is just very baby steps and early stages of trying to integrate this very complex spatial information that we get from our images. We're nowhere near what like ChatGPT can do, and these large language models, which are very impressive. And so I'm really excited about the potential of using more complex deep learning, neural networks, large language models, etc., in image analysis and making sense of these very complex images. So I'm excited, and we're trying to sort of dip our toes in a way in that field, but at very, very baby steps.
Yeah. So yeah, I like ChatGPT. So very powerful. So I'm not the expert, of course, but I've been talking with those people. And my impression, I like the discussion with one of my friends. His view is okay. The technical part is aside, so we need a lot of data, right? So without data, we just can't do work anyway. So the amount of work now increasingly, it's catching up now. So people start doing something good because of the amount of data is increasing. Our field is still, we are talking about how to increase the depth or throughput of for instance, spatial epigenome, 3D epigenome thing. It's just data is not there yet, in my view. So it's a two ways. One is increasing enough number, and then how to handle computationally, right? It's just storage. We just can't store the data, imaging data. So that's my. It's from the non-expert view, naive way to see, and then two arms, the technology, and then actual physical storage or just number of data that seems to be catching up now. So several years later, with you guys may be doing amazing things.
Yeah, first of all, I want to thank three of you for really teaching us how physical genomics is organized. And as someone who is used to looking at the DNA sequences and the chipsticks and all these things, I'm fascinated by this work. My question is more like directly to Melike. It's beautiful that you can just, with the right amount of softness, you can see that the changes in the chromatin. A lot of genes tend to go to the nuclear periphery when you soften the environment. So one thing is, we tend to think that the nuclear environment is a repressive region. Do you think generally genome is becoming more kind of suppressed when the environment is soft? And related to this one, do you think we can kind of use that information to engineer cells to sort of to go into a specific pathway with the right amount of softness or stress?
Yeah, those are all great questions. So, you know, we do see increased compaction, increased amount of chromatin at this, what is thought to be the repressive compartment near the nuclear lamina. So the expectation is that there's changes at the gene expression level and likely, you know, silencing of genes. Although, you know, that sort of relationship is very complex. So in the past, people have done experiments where they, just through tethering, you know, pulled genes to the nuclear lamina or from the lamina to the interior, and sometimes that's sufficient to activate or repress a gene, and other times it's not. And so I think just localizing something to the lamina does not necessarily mean that it's going to be silenced. So we need to do sequencing, which is something my collaborators are currently heavily working on to figure out which genes are affected. We see a very global remodeling. So we expect the effect to be also reflected globally at the gene expression level, but we don't know yet. So that's something that you know, our collaborators with RNA sequencing are following up on. So it would be very interesting to see the results of that. And then, yeah, so one of the questions that we have is, you know, especially with the disease context where the environment is softened, the cells reside in that soft environment, they remodel their spatial chromatin to a point where it's not reversible anymore, or at least in the time scale of our experiments, it's not reversible. Can we re-engineer that? So we are approaching it from, like, can we rescue maybe through changes in the mechanical environment, but also maybe chemical cues, right? Epigenetic cues, can we restore a more normal spatial organization? But you can also apply that type of thinking into, can we engineer the genome organization of cells in ways to direct them into different sort of functions? Yeah, absolutely.
Okay, I have lots of questions for all of you, but I wanted to ask one that I think is relevant for each of you. You all showed results that had sort of a chicken and egg quality. So for both of you, that's a little bit on the pathology, and for you, that's maybe a little bit more on the relationship between underlying truth and the pupil functions that you're extracting. And I'm really curious about your opinions on the role of this direct imaging in understanding, like, the causality. Why is something reversible, or is it that cancer is rearranging the chromatin, or is it that in a cancer cell the chromatin arrangement gets messed up? And what can your pupil functions tell you about that tissue in between? That's super broad, I know, but I guess I just wanted to give each of you a chance to talk a little bit more about that duality there.
I think that is a wonderful question, and one that, you know, keeps me awake at night all the time. What is the cause and consequence relationship, at least in my case, between the spatial organization of chromatin and gene expression, for example, or disease phenotype? And I think it's a very complicated, and in a few situations where we try to dissect that, it's both ways. So in some cases, genome organization can direct gene expression. In other cases, gene expression actually directs genome organization. So it's a two-way relationship, and it's very difficult to detangle. I think imaging has a good role in addressing this question because it's single cell based, right? So we can, and for example, you know, the heterogeneity of senescence, I think single cell tools are very powerful in looking at heterogeneous samples, and then sort of with multimodal imaging, right? Like layering multiple types of information on top of each other, which is another thing that imaging tools are good at, not just DNA, but you can look at RNA and, you can look at proteins. So you can sort of combine all that together with more engineering tools where you can manipulate, you can just manipulate gene expression, for example, with these CRISPR-Cas9 crab domains, and then look at the impact on all the other components. I think we can start to get at this cause-consequence relationship. So we need to combine, I think, engineering, imaging, multimodal imaging, and disease models to address that. Yeah, but we're not quite there yet.
Yet, that's a fundamental question. So the imaging, if we have super great imaging, we're not going to do high-C anymore. Right? We cannot do this. That's why we have to do this kind of an indirect, so tedious thing. And then so many steps there, and then we have there. Right? Any mistake, and it could distort data. It's kind of scary. If you can see by eye, why should we do this kind of thing? So it's not going to, in the near future, right, waiting for you, no one would do high C. So, I mean, the imaging, I mean, together with deep learning, right, so no more such tedious experiments in relatively soon, near future, is my view. We don't work, I mean, I showed some cancer, but we don't work on cancer much, even though I'm a cancer biologist, I see myself. We work on pre-neoplasia, or the reason why, because still there are lots of physiological rules there, right? So it's very much easier to extract the data information. Compare between normal and cancer, it's really tough thing because it's just completely different species. So step by step, right? So pre-neoplasia, tumor initiation, and then what's the next step? What's the next step? That's how we want to do. So that's a trajectory, like short span, longer span in a 3D epigenome trajectory. If we can do, I mean, that's a lot of things we can learn. Stochastic, and basically, what's that? Why stochastic? And that's Joanna can maybe have much better idea. But what's the stochasticity? I mean, and if you can visualize, oh, this guy or that guy, that's going to be I think it's the future.
Maybe I can warp this question a little bit for both of you, right? We have the question of can we resolve chromatins and really high accuracy, and so that we don't do high C. But Maliki already showed beautiful data, and potentially, what's the challenges that you can develop a new methodology to replace high C?
So, I mean, I think we already have technologies like that, like spatial genomics and spatial transcriptomics, right? Again, we're in the early days. I think the throughput is still low. So throughput is one big issue that we need to overcome. The coverage, we still, I think, need to compromise within a number of genomic regions we image versus the resolution and being able to see the folding and the because nucleus is very crowded, right? So expansion microscopy, which Marcus in my lab brought to my lab, I'm very excited about combining that with some of these spatial omics technologies. But I think, we are starting to get there, but, we still need more developments in these fields to completely replace something like high C. We're not there yet. Yeah.
Amazing, yes.
Hi. Really great talks. I enjoyed everything you guys have shared. I have a specific question for Dr. Lakadamyali. In your work, I was intrigued by the mean variance relationship in the clutch size, where the samples that had a larger clutch size also had more variation. I was curious if you could explain why there is that mean variance relationship, and if you think that the variance is important for the effect on plasticity that you guys have observed.
Yeah, that's a wonderful question. So the samples that had the larger sort of clutch size were the more sort of differentiated states, less pluripotent cells, which also, when you take ES cells and you induce differentiation, there's a lot of heterogeneity in that process. And the end state, some cells do differentiate, others don't. So I think that's where the heterogeneity or the variance is coming from, is the variability in the cell state itself. So some cells are in a more terminally differentiated state or progenitor state, versus other cells are more in a ES state, and that's where the cell-to-cell variability comes from. I forgot what was your second question. I think there was a second question.
I was wondering if you thought the variation was important for the pluripotency effect.
Oh, that's right. Yeah. Yeah. So again, like the cause-consequence relationship is still, not clear. Is it the cell fate changes that is leading to the changes in chromatin, or the changes in chromatin that is leading to cell fate changes? So it's hard to address that. But I think, in terms of the variability, that's real variability in the cell state that we're seeing. Yeah.
Yeah. I have one question for Dr. Narita, specifically. Thank you for excellent talk. Quick question is that you mentioned that not only histone, but also non-histone HNG-A1 is required for the heterochromatic foci formation in senescent cell. So as the SSP, the SASP or SMS are cell type specific. Do you think that HMG-A1 is required for heterochromatic foci formation for all sorts of cells or only in fibroblasts? Did you check in the endothelial cell or cardiomyocyte also go sometimes? It can undergo senescence, although they are terminally differentiated. Right. So my question is whether it is required for all sorts of cells or not. So thank you.
That's a very important question. So the senescence, many people using senescence, but it's really classic senescence is quite limited to the human diploid fibroblast, and then from there, because it's so heterogeneous and it's so diverse, so the fragmented information are applied to the other different things. So to me, it's many like senescence-like phenotypes. It's a senescence-like phenotype. It's not. We don't know. We don't have the clear definition. That's why our new preprint, the research square, address that kind of question in a specific in vivo model. So. But having said this, so the last, even in a human diploid fibroblast, I showed the heterogeneity, right? So not all cells do show SAF, for example. So that's probably correlate with, at least in part, with the quantitative level of HMGA level. So it's a kind of HMGA provides additional flavor of what kind of senescence. And then the SAS, you asked how HMGA affect the SAS. So I skipped that slide. So so HMGA sort of promote condensation, but at the same time excluding the euchromatic regions, right? So this is coupled. But important thing, if you remove HMGA, the condensation largely go back to the original. But this guy, okay, many of them go back to the original thing. But some enhancers, like SASP genes enhancer, don't come back. They enhance. They we see the de novo interactions. Okay, this is in the context of oncogenic RAS, so still oncogenic RAS in this sense phenotype. So that's why what I meant, like a buffer activity. So HMGA, for some reason, right? So SASP goes up during senescence, but if you don't have much of HMGA, even higher. So HMGA is sort of buffering this one. This is connected to the other fibrogenic activity, for example, or some chromatin compartmentalization. So how universal it is, it's still, we need to look into that. But that part is quite generalizable because it's not even senescence. We can look at, I don't have time to show the data, but cancer cell data, right? So HMGA, high, low HMGA have a big impact or same direction on the inflammatory cytokines. So I think this is far even beyond the senescence regulation. Heterochromatin part is quite unique to senescence. We don't know why. That's the future, one of the future direction we are working on now. Sorry, so I probably wasn't super clear.
Thank you. So we have time for one more urgent question. If you do have it, raise your hand. I'll pass you the mic. Let me ask you. So in your talks, in your careers, you were able to break several boundaries and show that some things that everybody knew, or presumably quote unquote knew, were correct, turn out to be not exactly correct things. And we saw some examples of that in your talks. Are there some well-known facts that most people believe in and take for granted that you think are going to be proven to be wrong in the next several years?
I can speak obviously.
I already said that.
Maybe I can start with a light one so that you guys can go heavy. Maybe some of you agree, and I would say one of the papers I talked last is currently accepted, and we have a derivation that numerical methods does not increase resolution. It can only keep your resolution flat or worsen it. And this is only applied to deterministic numerical analysis. If you put randomness in your numerical analysis, you create noise and potentially get worse. So that is proven in the supplement. I hope you can go in and read it. We prove that in the information theory.
I think I gave already a few examples. So HMGA biology. So HMGA is not so popular, right? So everybody knows nucleosome, histone code. Not many people don't care. So I think that's wrong, right? You have to be more interested in that because it has a big impact. And now before the sequencing era, I mean, I don't say those are wrong, right? Those are correct in that kind of a in vitro recombinant protein synthesized DNA, what's happening. We are also doing the phase separation. I think the data correct, but I'm not confident how much the HMG phase separation is involved in the cell formation. I don't have any confidence yet. It's a great concept. We are going that direction, but I'm skeptical myself. Right, so now and it's not so popular. No one is really working much. So even after the sequencing available, not much advanced there. Right, so it's in vitro, pure model dependent truth or correctness, and then actual real world correctness is a different things. Right, both are correct, but just simply in a different system. So I think that's where we are, and I highly recommend to be more conscious about HMG-A, not B. Oh, okay, I'm kidding. A. Yeah. So.
Yeah, I don't really have a specific example, but there are probably trainees in the room. I encourage them to challenge, the textbook models, because often those models, are just cartoons. And then when you dig into sort of how we came up with those cartoons, you realize that, the evidence maybe sometimes is incomplete, other times maybe outdated. So, and I think this is why it's important to get people from different backgrounds, different fields, and different perspectives together. Again, when I came into chromatin field, I was pretty ignorant. I didn't realize the importance of this 30 nanometer fiber. And in a way, that was perhaps a good thing because I didn't take it for granted. And I think it's important to look at things from different perspectives and question, some of the cartoons that we like to draw to simplify things.
Right, and I think your example of 30 nanometer chromatin fiber is a good one because it has been very popular in all textbooks. I think it's still on Wikipedia, last time I checked, so apparently accepted. So the bottom line is, don't be afraid of challenging what textbooks say. After all, there was a time, as we all know, when Earth was believed to be flat in the center of the universe, right? It's not exactly flat, right? It's kind of ellipsoidal, a little curvature on it. Well, thank you so much. This was wonderful. Thank you for your beautiful talks. Thank you for your contribution.
Thank you, Vadim. Thanks.